All right. All the rest of this stuff is review. Please make sure you know this. If you don't know this, it's not going to be good. Right. So I'm going to go through this pretty quickly. You're going to read the book. You're going to know all of this. It's 107 content. There is no excuse for not knowing all of this content. It's a prerequisite for this course. So if you don't know this, you better get on your horse. Okay. Ribose and deoxyribose, these are what? The sugars that are in our nucleic acids, right? Ribose contains an oxygen, or a hydroxyl group, at both the 2' carbon and the 3' carbon. All nucleotides have a hydroxyl at the 3' carbon. That's the 3' OH I was constantly yammering about in 107. The 5' carbon is the one that the phosphate group is attached to, right? 5' phosphates, 3' OH. These don't change between these two sugar molecules. Just the 2'. Deoxy, remove the oxygen. So when you're looking at pictures of nucleotides to determine if they're DNA or RNA, make sure you're looking at the 2' carbon and not the 3'. So don't just say this and go, oh, look, an oxygen has to be ribose. You have to look at the 2'. Okay? So ribose has 2 hydroxyls, deoxyribose only has 1. And then, of course, we need to know the purines and the pyrimidines, and you need to know which class is which, right? Adenine and guanine are both purines. They have the 2 rings. Cytosine, thymine, and uracil are all pyrimidines. They only have the 1 ring. I won't ask you to draw the structures as far as carbons and hydrogens and nitrogens and oxygens or any of that kind of stuff, but you do need to be able to recognize them as pyrimidine or purine, just like you did for 107. Oh my God! That is a giant phosphate. All right, so if we're looking at, excuse me, the 4 nucleotides of DNA, and again, how do we know they're DNA? Right? 2' is missing the hydroxyl. And if these were in a category, we'd automatically see that this is thymine, right? Deoxythymidine, which means this is the T, right? And there are no T's in RNA, so this would have to be DNA. So here's our C, here's our G, and here's our A. And as we know, this is just the monophosphate, right? Which means it has 1 phosphate group. We do know that they exist each with 2 more phosphate groups attached as a trinucleotide. So if we were talking about adenine and it had its 2 extra phosphate groups floating around, right? This is ATP, people. DATP in this case. And the D just stands for deoxy. Okay, make sure you can recognize these structures. And then, of course, our beloved double-stranded DNA. I'm going to point out the things that I just cannot let you guys get wrong, right? We're first, right here, no oxygen in that 2'. The 3' is the one that, right, connects. What is that bond called? Phosphodiester bond, right? Between the 5' phosphate and the 3' OH, right? 5' to 3' direction, right? 5' phosphate hanging off one end, 3' OH hanging off the other. On the other strand of this double strand must be anti-parallel, right? In this case, the 5' down here and the 3' OH here, right? We call those 2 strands together anti-parallel. That's what sets up this hydrogen bonding between the bases, right? We have to remember we've got phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, and the bases are what are attracted to each other by hydrogen bonding. And that's DNA, always, always, always double-stranded, except very transiently during mechanisms. Okay. If we look at RNA, it's always single-stranded, but again, 5' to 3', right? Phosphate, sugar base, phosphate, sugar base, phosphate, sugar base. And here we can see, right, here's our telltale extra hydroxyl group, which tells us it's RNA. If we look at DNA in the space-filling model, we can see that the backbone is what we can see that's spinning around and around. Okay, the double strand in here, where the base pairing is, and the way it's twisted, there's a major groove and a minor groove. Okay, this is just a little picture that's showing us this. Again, 5' to 3' on one end, right? 5' to 3' anti-parallel, base pairing down the middle. I don't care about all these distances. That is, you can always just look that up. So don't memorize distances. Make sure you know about anti-parallel.